Life is made up of 6 major elements: carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus, which are all complemented by a selected array of trace elements such as calcium, iron, etc. There are many scenarios in the past of trace elements substituting for one another. However, there have been no prior reports of substitution with the 6 major elements. This particular article presents a situation where a trace element, arsenic, replaces phosphorus in macromolecule formation in living organisms. Specifically, the macromolecule they focused on was nucleic acids, such as DNA and RNA. Nucleic acids are polymers made up of monomers called nucleotides. There are 3 components of a nucleotide: the phosphate group, the 5-carbon sugar, and the nitrogenous base. Ribonucleotides are the monomers of the nucleic acid RNA, and deoxyribonucleotides are the monomers of the nucleic acid DNA.
The scientific article we will be focusing on, a bacterium that can grow by using arsenic instead of phosphorus, headed by Felisa Wolf-Simon, focuses on how a certain bacterial strain found in Lake Mona, called GFAJ1, can thrive in an arsenic-rich environment. They hypothesized that an arsenic group could specifically substitute for the phosphate group on nucleic acids. The scientists isolated a strain of GFAJ1 and set up 3 artificial media: an arsenic-rich media without any added phosphorus, a phosphorus-rich media without any added arsenic, and the control media deficient of both arsenic and phosphorus. The isolated bacterial strains were placed in each of these media under regulated pH and temperatures to be observed for a period of time. The scientists found that in the arsenic-rich media, growth was observed in the bacterial strain. Meanwhile, in the phosphorus-rich media, growth was also observed in the bacterial strain, but at a faster rate. However, in the control media, no growth was observed. Finally, the total intracellular phosphorus in the arsenic-rich medium was consistently far below the quantity needed to support growth after the experiment was repeated several times. With these results in mind, here are some comments on the research conducted by Wolf-Simon.
Comment. Rosemary J. Redfield. Methodology. The phosphate level present in the contaminated growth medium would have provided enough phosphorus for all the cell growth seen in the arsenic-rich medium. Also, further steps to minimize contamination were not put in place to ensure accuracy. The steps used for treating the DNA and RNA pellets were substandard, and this calls to question the validity of the results from this experiment. In summary, the methodology of this experiment is being called to question.
In response to Redfield, Wolf-Simon mentioned that the cells were collected by centrifugation and triple washed in the same mineral salt medium that they were grown in. There was no additional glucose, vitamins, arsenic, phosphate, or trace metals. The cells were then put through the normal DNA-RNA protocol, removing any unincorporated arsenic impurities. The DNA-RNA pellet was collected by centrifugation and washed with 70% ethanol, then dried and suspended in ultra-clean water. The scientists believe conducting the experiment in this manner shouldn't cause a significant contamination of phosphorus in the DNA sample.
Comment by Kottner and Hall. Wolf-Simon cited Kottner and Hall's work done with E. coli and their phosphorus content. This is not a good comparison because they were cultured on high phosphate levels and their growth rates are higher than typical bacteria in the environment. In addition, Kottner and Hall recently published a survey on the elemental content in 120 freshwater ecosystems. Their findings show that the phosphate content of aquatic bacteria samples can be highly depleted of phosphate. For example, one ecosystem had a mean of 0.5% phosphate and some individual bacteria had 0.03% phosphate. So the low phosphate levels reported in Wolf-Simon's experiment are not uncommon. Since they found low growth rates in Wolf-Simon's experiment, the data cannot also be used to support the claim that arsenic can substitute for phosphate.
In response to Kottner saying their data cannot be cited, Wolf-Simon claims that if you compare the population average instead of the phosphorus content of the community, this can be relevant to our experiment. In both experiments, the phosphorus levels are normal or ultra low, so they're insignificant to the experiment.
Conclusion. This topic is still open to research and investigation by a scientific community. There has been no strong conclusive evidence on why and how the bacteria strain GFAJ1 was able to substitute arsenic for phosphor in their nucleic acid. Most of the original scientists who worked with Wolf-Simon on this experiment have moved on to new ideas. But scientists around the world are still trying to expand this ongoing research because samples of GFAJ1 are now available to the scientific community.